Thank you very much for inviting us to talk today. I'm the Chief Scientific and Chief Medical Officer at Therapeutics. We are a San Francisco and Berkeley, California-based biotechnology company, and it's a pleasure to give you an update on our progress in 2024, where our mission is to restore vision to those patients with severe retinal degenerations using optogenetic therapy. We have lead assets, RTX-015 and 021, and I'll go into a little bit of detail about those in future slides. But suffice it to say that these are the first bioengineered optogenetic medicines optimized for human vision, and they have the capability now to restore higher visual functions without the need for goggles, which have been used for most of the optogenetic clinical trials that have been undertaken to date. We are a small company, but we have a very experienced management team, particularly in the optogenetic space and AAV technology. And we believe, based upon the work that's been done over the last 10 to 15 years in optogenetics for vision restoration, that we have a very de-risked program, because essentially optogenetics does work to improve some vision in patients. That's been clearly demonstrated. And then when we look at what our assets are able to do, it allows us to develop a clear first-to-market strategy with that. It's been a busy year for us in terms of funding. We have raised $114 million in the last couple of years, most of it in this year, and we're extremely grateful to the top-tier investment groups that have funded us in our Series A back in May. We're also very grateful to the funding from the California Institute of Regenerative Medicine, which has really allowed us to pump prime our assets in preclinical development. So just a word about where does optogenetics fit into the program. And we've heard some absolutely excellent talks so far, developing treatments for retinal degenerations, primarily where you have existing photoreceptors. Optogenetics is different in that there's a significant proportion of the patient population with retinal degenerations, or in many cases RP, where many of their photoreceptors have already been lost, and most of these new treatments won't be able to target those because those cells have been lost, whereas optogenetics is uniquely almost able to restore vision, or has the potential to restore vision, in those patients who've lost significant amounts of vision. Just a quick word about optogenetics, which I'm sure many people are familiar with, but it's based around observations in single-cell microbes that they can move towards light or away. And it's the way they do this is they have these special, unique, in fact, channelrhodopsins on their cell surface, which are light-gated ion channels. We can harness those. These are proteins which can be encoded genetically and expressed using an AAV in retinal cells, and reanimating those retinal cells that survive in retinal degenerations like retinal ganglion cells to receive light and respond to light with an electrical signal. And although it's a very disruptive and sophisticated technology, primarily employed in the neuroscience field, it's been around for about 20 years. And we really feel that it stood the test of time. It's now maturing as a technology, and here we are really beginning to see the fruits of our labors in visual optogenetics now. Several programs have reported positive results. One that really has hit the sort of top-tier journal publications was from a group called GenSight, where they saw a profoundly blind individual restored vision using optogenetic therapy when wearing the goggle systems. But nonetheless, really a lovely proof of concept. And then hopefully here we come at Therapeutics with something which has been truly bioengineered for human vision, allowing greater and better outcomes. So our lead indication is in retinitis pigmentosa, and we have funding also to move forward into Stargardt's and geographic atrophy with our other indication. Just a brief overview of how this technology works. Intravitreally delivered AAVs, as we've heard from others, this is a real workhorse vector approach. We use a sophisticated variant that allows retinal transduction at low doses. In the case of RTX-015, it's targeted towards retinal ganglion cells. In the case of 021, it's targeted towards bipolar cells. And I'll come on to the reasons for that in just a second. Here's some of our data. In terms of the bioengineering, it allows us to bioengineer something that's really light sensitive, so light sensitive right through the human cone opsin range. And compared with the first generation optogenetics, we're about 1,000 to 100 times more light sensitive and therefore won't need goggles. And also because of the sophisticated bioengineering, we're able to see higher visual functions appear. This is a mouse study showing that we are able to achieve at least 50% visual acuity creation in previously blind mice and near normal levels of contrast sensitivity with RTX 015 for ganglion cells. When we do the same studies, we think we're sort of getting a ceiling effect with 021 in terms of visual acuity, but you can see much improved light sensitivity and improved contrast sensitivity going after those bipolar cells, which once again, when we think about the diseases, RP, primarily the retinal ganglion cells survive late in the disease. In other IRDs and geographic atrophy, then we know the bipolar cells are present. And that really allows us to think carefully about our value proposition. So for the first time, we have a human channel rhodopsin, which is optimized for, like human opsins, for light sensitivity, and that we're seeing a wide dynamic range and highly sophisticated switching, which allows for this higher visual functions to be a realistic possibility. Encoded as an AAV-based gene therapy with 015 for retinal ganglion cells and 021 now for bipolar cells at low dose, giving a safe, durable gene therapy benefit, and as we've heard from others, a really attractive commercial opportunity. So those are our programs. We hope to be in clinic early next year with our lead asset, RTX 015. We've taken all of the necessary steps to de-risk the manufacturing. We're going in with a commercial process from the get-go to allow us to scale up and move rapidly through clinical development. One of the really intriguing things about optogenetics is from a clinical trial perspective, the safety population suitable for phase 1 development is in fact the treatment population. And so we should be able to see efficacy early on in our approaches. And with that, thank you very much indeed for your attention. Thank you.